So welcome everyone, and thank you for joining us virtually for the latest of the Variant Effect Seminar Series. My name is Jan. I'm a member of the VES Organizing Committee, along with Adrini de Souza, Diego Calderon, Ohana Bezeres, and Mariano Martin. These seminars are made possible with a lot of support from Lara Muffley and Alex Hopkins, among several others, and our sponsors of the Atlas of Variant Effect Alliance. As a reminder, we're always on the lookout for potential speakers for future seminars. If there is anyone you would like to see present, or if you're interested in sharing some of your own work, then feel free to submit a speaker nomination form. As usual, before starting the presentation, I have a few housekeeping items to mention. Each talk is around 20 to 25 minutes, but there should be time for a few questions. Please post your questions in the Q&A. We'll read them out loud to the speaker at the end. If you have additional questions at the end of the seminar, or if you're an AVE member, feel free to visit our seminar series Slack channel for more discussion. And finally, we're posting updates on our Twitter account, so be sure to follow us there. You can catch today's seminar and previous presentation that have been recorded on our affiliate CMAP YouTube channel. So don't worry about trying to copy the URLs. They will be posted in the Q&A in the chat channel here. All details of this seminar series can be found on our website, including a regularly updated schedule of the upcoming speakers. For the last housekeeping items, this is rarely an issue. Please be considerate when posting comments or questions. We expect everyone to be respectful and to use inclusive language. So without further ado, the first speaker is Nadav Brandes. Nadav obtained his PhD in computer science from the Hebrew University of Jerusalem, and is currently a cancer research institute and European Molecular Biology Organization postdoctoral fellow at the University of California in San Francisco. His research focuses on the development of machine and deep learning approaches to study the effect of genetic variation on human traits and disease. Nadav, without further ado, you can take it away.
Thank you. Thank you for the introduction. Okay, can you see my slides?
Yes.
Okay, thank you. Yeah. I want to start by thanking the organizers for inviting me to give this short presentation. And I also want to say that I really like this seminar series, and I also really appreciate this community and all the great work done here. So many of us in this community are interested in variant effect prediction computationally. So given a specific mutation predicting, for example, whether it's pathogenic or benign. And I want to say upfront that throughout this presentation, I'm going to focus on coding effects, on mutations that change the amino acid sequence of a protein product. But I think that the key ideas I'm going to explore and share with you today could, at least in principle, be generalized to non-coding effects as well. So throughout the years, people have developed many methods to predict variant effects, and you can see here a small subset of all the different methods that exist out there. We have supervised methods and unsupervised methods using all kinds of features and sources of evidence and training algorithms. And what all of those methods have in common is that they were developed specifically for the task of variant effect prediction. And because of that, they have limited generality, because the common wisdom is that we can only expect methods to apply for the types of mutations that they have been trained on. For example, most methods will only work for missense mutations, even though there are many important mutations that are more complex, like in-frame indels and early stop gains that could have important clinical effects. And also, there is very little work done on providing different predictions in the context of alternative protein isoforms, even though alternative isoforms are a pretty important feature of human genomics. And finally, most methods are dependent on some form or another of external information beyond the protein sequence itself. So, for example, many methods will depend on multiple sequence alignment and regions that don't have sufficiently close homologs will not work very well. So most of these limitations, like I said, they are the result of the fact that that's simply how we classically do machine learning, by training models on the specific tasks that we intend them to perform. But over the past years, we've seen a different paradigm coming to the forefront of AI, where instead of training models on the specific tasks that we intend to use them for, we can train far more general AI models known as foundation models. And perhaps the most obvious example of that is large language models, which can perform a really wide array of tasks, from answering simple factual questions to writing programming code. And with recent image support, you can now upload an image and ask questions about it, or put a description and let it generate images. And the underlying algorithm that has enabled so many of these impressive applications is actually embarrassingly simple. So large language models are trained on a super simple task, which is just predicting what's the right word in each position in a sentence. And by training sufficiently large neural networks on sufficiently large collections of human written text, those models are able to absorb a lot of underlying knowledge about human language and about the world, which enables all of these impressive applications that we're now seeing. So we have an opportunity to do variant effect prediction using this new AI paradigm. So instead of training models specifically to perform variant effect prediction, we can train models on a more general task. And as I'm going to show you today, that's going to enable a more general model for variant effect prediction that, for example, would work not just for missense mutations, but any coding mutation in any amino acid sequence, and would be able to provide different predictions in the context of alternative protein isoforms, and will not need any additional information. So throughout this short presentation, I'm going to talk about protein language models, which are simply large language models trained on protein sequences. And I'm going to show that those protein language models can predict variant effects very accurately. I'm going to show you exactly how I evaluated and compared that approach to other methods, and how we could generalize them not just to missense mutations, but also to more complex mutations, and also deal with alternative isoforms. So like text-based LLMs, protein language models are trained on this super simple task, which is predicting what's the right amino acid at each protein position. And again, by training sufficiently large neural networks on a sufficiently large number of proteins, we can enable those models to learn a lot about proteins. And even though it's a very recent approach, there is already vast literature showing that those models can indeed learn a lot about protein structure and protein function, and enable very efficient prediction on almost any protein-related task. For example, here you can see a figure from an earlier work of mine where I show that by pre-training the model on the protein language modeling task, it also improves at predicting secondary structure. Here, there is a pretty moderate but very consistent improvement from about 70% to about 74% accuracy. But of course, the exact details will depend on the specific task and model. In the work I'm going to show you today, we used ESM, which is a fairly large protein language model with 650 million parameters. It's trained on all 250 million protein sequences in UniProts. So given the protein sequence, ESM will train by masking random residues along the protein sequence, and then allowing the network to predict what's the right amino acid at each protein position. So at each position, we predict the distribution over all possible amino acids. And by comparing those predictions to the input sequence, over time, the network will get better at predicting amino acid sequences. So the output we get from the model is the amino acid substitution probability matrix. So again, at each position, we'll get a distribution over all 20 standard amino acids. So in other words, we'll get the likelihood for every possible missense mutation along the protein sequence by training the model to predict the sequences of proteins selected throughout evolution. Now, one of the main forces of evolution is negative selection, where mutations that cause a disease or otherwise disrupt the function of a protein would be selected against. So this naturally gives rise to the hypothesis that we expect damaging mutations to be predicted as less likely. And that's exactly what we observed. We compared the distribution of ESM scores assigned to mutations expected to be mostly pathogenic and mutations expected to be mostly benign, and get very good separation between these two distributions with an overall AUC score of about 90%. And we compared ESM to 18 other variant effect prediction methods on two different clinical benchmarks using two slight variations of the AUC metric. And in all cases, we see that ESM outperforms all other methods. When I first saw that, I was actually quite surprised because those other methods are actually very strong methods, and there was a lot of effort and engineering ingenuity going into developing them. Whereas ESM is a very general protein language model that wasn't even trained with a specific task in mind. And still, we could take the model exactly as it is without fine-tuning or changing anything in it. And just looking at the probabilities assigned by ESM, we get variant effect predictions that outperform all of these highly specialized methods. When we do these comparisons, we need to be aware of data circularity concerns. So sometimes the same evidence, specifically allele frequency, can be used both as features for these methods and also as one of the criteria for labeling variants as pathogenic or benign. And some of these methods are actually directly trained on such clinical data sets. So in the comparison I just showed you, we excluded any method that either used allele frequency as a feature or was directly trained on such clinical data sets. But then how can we compare against methods that violate these criteria? So for that, we need a different benchmark, and we use deep mutational scans, which measure various cellular phenotypes as proxies for the clinical outcomes. For example, we could measure cellular growth or proliferation as a proxy for cancer. And because most methods aren't directly trained on this data, we don't have the data circularity concern. And in this case, we could compare ESM to any other method that we knew existed out there on this benchmark of 28 different assays in human. And again, we saw that ESM outperformed all other methods. Now, there are actually multiple ESM models that exist, and in this work, we specifically used ESM1B, but I know that others have used other variations of ESM, specifically the ESM1V models. But when comparing on these specific benchmarks using the model in the specific way that we did to predict variant effects, we actually saw that ESM1V slightly outperforms the other ESM models. And it's only if you average the predictions of multiple ESM models, you can get slightly better predictions, which is expected. It's typically the case that ensemble models can slightly outperform base models. So everything I've showed you so far deals just with missense variants. So to recap how we score those variants, we simply take a protein sequence, we feed it through ESM without changing the model in any way. We get the probabilities from the model, and then to score a specific missense variant, we calculate the log likelihood ratio between the probability that ESM assigns to the mutated amino acids and the probability it assigns to the wild type amino acid at that exact position. But like I said, there are many other types of important variants beyond just missense mutations, including, for example, in-frame indels that can introduce arbitrarily complex changes to the amino acid sequence. So can we use ESM exactly as it is without fine-tuning it to score those more complex types of mutations as well? So the key idea to do that is that if we can somehow take the probabilities given by the model to assign a likelihood to any given sequence, then we would be able to score any coding variants by, again, calculating the log likelihood ratio between the probability assigned to the entire mutated sequence and the probability assigned to the entire wild-type sequence. So how can we do that? If, again, we look at the probabilities given by ESM, then at each position, some of the probability mass will be assigned to the amino acid that is given as input, whereas the rest of the probability mass will be distributed between the 19 other amino acids. And the more likely the specific amino acid appears to ESM at that exact position, the more of the probability mass will be assigned to that specific amino acid. And if we assume independence between positions, we can simply multiply all of these probabilities between positions or summing them up in log scale to get a pseudo-log likelihood for the overall sequence. And now to score any given coding mutation, we can simply calculate a pseudo-log likelihood for the mutated and the wild-type sequence by fitting them separately through ESM and then subtracting the two numbers. So again, we tested that on CleanVar by looking at in-frame indels labeled as pathogenic or benign, and again, get very good separation with an overall AUC score of 87%, which is quite close to the 90% we get for missense mutations, and compares favorably to another method capable of scoring such mutations and some baseline models. To deal with stop-gain mutations, we took a slightly different approach, and we want to do that based just on invoking the wild-type sequence, which would be much more efficient. So the kind of intuition here is that the more important the region that is deleted at the end of the protein sequence, and as a result of the new stop-gain codon, then the more likely it is to be pathogenic. So to score any stop-gain, we simply look at the missense mutation among all possible missense mutations in the deleted region and take the mutation predicted to be most damaging and associate a score for the stop gain with the score of that missense mutation. And again, when we look at mutations that are not expected to undergo nonsense-mediated decay according to the 50 base pair rule, then we get pretty good prediction performance for whether mutations are pathogenic or benign. But for mutations that are expected to undergo NMD, we get very poor performance, which makes sense because NMD happens at the transcript level and protein language models have information only about what happens at the protein level. And sometimes even just looking at the variant effect predictions at very low resolution can show interesting patterns about proteins. For example, we can see which regions are more sensitive to missense mutations and, more sensitive and which ones are less sensitive. And it appears that those regions predicted to be most sensitive very often overlap with known protein domains. And that's true also for regions that are not covered by multiple sequence alignment, which again highlights the advantage of protein language models compared to homology-based approaches. So I want to wrap up by talking about alternative protein isoforms. We know that due to alternative splicing, the same gene can result in multiple protein isoforms that can have different regions of the proteins either included or excluded from them. And it seems very plausible that the effects of a mutation will depend on the context of the specific protein isoform. So for example, we could have a mutation that is damaging to the primary isoform, but because of interactions with other regions of the protein, it will be neutral in other isoforms or vice versa. And because protein language models can just look at the amino acid sequence of each protein, and they don't depend on any external information such as homologs, which will often be shared between isoforms, they can really interpret mutations based on the specific amino acid context of each isoform in a relatively unbiased manner, which gives us this opportunity to study different effects of mutations in the context of alternative isoforms. So we want to see how common it is that ESM assigns fundamentally different predictions to the same mutation in alternative isoforms. So we define a variant to be isoform sensitive if it's predicted to be likely neutral in at least one isoform with a score above -7, and likely damaging in at least one isoform with a score below -8. And the difference between the maximum and minimum score is at least 4. And we found almost 2 million such isoform sensitive variants. And in fact, they exist in most genes that have more than one isoform. And as expected, we see that isoform sensitive variants tend to be closer to splicing junctions. And we also see that genes with isoform sensitive variants are more likely to have domains disrupted by splicing. So I mean, for example, cases like the green domain you see here in the second and fourth isoform, where you end up in this awkward situation where only part of the domain is included, as opposed, for example, to the yellow domain, which is either included intact or missing entirely. And I want to finish with a nice and interesting example of a gene called MAN1, which is a tumor suppressor. And in addition to the primary isoform, it has a second isoform that is very, very similar to the first isoform, except of a very small deletion of only 5 residues. But when we compare the effect scores assigned to missense mutations between these two isoforms, we see that there is a much larger region that seems to light up. And we in fact see that there is an entirely new region that only seems to be functional in the second isoform. So to gain some intuition about what might be happening, we looked at the predicted structure of these two isoforms according to AlphaFold. And we see that in the second isoform, there appears to be a small pocket on the surface of the protein that all of a sudden gets exposed when those 5 residues are deleted. So this might be some form of a binding site or another active site that is exposed and only exists in the second isoform, which could explain those isoform sensitive variants. I think I'll skip this example. And yeah, finally, I want to say that if you want to use the pipeline that we developed, you can go to our GitHub, and we have command line tools and Python scripts for scoring any type of coding mutations, either missense mutations or more complex types of mutations, given any amino acid sequence that you provide. And you have the link here and also on our paper. And if you're just interested in missense mutations in human, then you don't even have to run our scripts. You can just go to a web portal that we developed on Hugging Face, and you can browse and download the effects of all possible missense mutations in all existing protein isoforms in the human genome. And again, you have the link here and also on our paper. So thank you so much for listening. I want to thank my founders, and I want to thank all my collaborators who work together on this project. And I want to especially thank Jimmy, my postdoc advisor, and to Vasilis, who has his own independent lab at UCSF and was leading this project. So to summarize, we've seen that large language models are state-of-the-art predictors of variant effects. They're very general and flexible, and we've seen that with respect for them being able to work with any coding mutation, not just missense mutations, but also mutations that affect multiple residues. We have a single universal model that applies to any amino acid sequence you provide it, and it doesn't need any additional information and can provide different predictions for the same mutation in the context of alternative protein isoforms. And it's actually more because of this incredible generality, even more than the sheer performance, that I'm very optimistic about the long-term impact of this approach for variant effect prediction. And I'll be more than happy to take questions.
Thank you, Nadav, for the amazing talk. It was very interesting. If anybody has some questions, you can just describe it in our chat. And while we wait to see if we have questions, I have one question. I think it's interesting that you're using like this model for predicting variant effects. And I imagine that you're using, as I understood, you're using like the amino acids, right? What you're reading, it's the amino acids of the protein.
Yeah.
So I was wondering if you can use this for predicting variant effect, you can also use it for predicting like the functions of the protein, right? So I'd like to know more about how accurate it is for predicting like characteristics of a whole protein.
Yeah, exactly. Actually, that's what people originally used protein language models for. It was kind of like, I think it took like a couple of years before people understood that those probabilities that they give could also be used as a very strong predictor of variant effect. Initially, the idea was just to absorb a lot of information about proteins, and then fine-tuning the models to predict various aspects of protein structure and protein function. And the early works on protein language models actually are pretty exclusively exploring that. So once you pre-train the models to do this protein language modeling, then you can fine-tune them with much fewer data on various tasks. For example, predicting secondary structure, which is the example I showed at the beginning, but you can also predict other aspects of protein function, like post-translational modifications, finding homologs, and also predicting structures. So for example, the model I showed you, ESM, it has an extension called ESM fold, which builds on this pre-training as a language model to predict protein structure. And it has many advantages over kind of like more complex models like AlphaFold, which are very heavily dependent on homologs. They are much slower to run, and they also tend to be a little bit biased towards the wild type in a way that protein language models, which only get the amino acid sequence of the specific protein you are interested in, are far less biased in this way.
We have some questions here in the chat. Matthew said, great talk and useful tool, thanks. Do you think the LLM might be using some level of MSA within uniprot proteins, for example, between paralogs and homologous?
So it's not doing that explicitly. Again, like what you actually give the model is just the amino acid sequence. You don't give it any other information. You don't give it multiple sequence alignments. But it could be that implicitly, kind of like while you train the model on uniprot, it does learn, kind of like this, kind of like memorize a little bit the space of protein sequences. So it probably has some notion of kind of like, which other proteins, like which other sequences might be similar to the sequence that you give it as an input. But it's not something you do explicitly. And there is also evidence that insofar as there is this kind of like memorization, it's not the whole story. So even if you give sequences that don't have very close homologs, it still works decently well. So there is, I think, by now strong evidence that those models, they don't just memorize the space of protein sequences that exist. They actually learn some aspects of protein structure and function more from kind of like first principles perspective, actually understanding the biochemistry of proteins.
Okay. One more question from Francesca. Where do you see the biggest challenge for protein language models for variant effect prediction?
Sorry, could you repeat the beginning of the question?
Where do you see like the biggest challenge for protein language models predicting variant effect prediction?
That's a good question. So I think really does work amazingly well. So it already works well, I think. So there is strong evidence that it works for missense mutations and more complex mutations. I think it's still to be seen, like how far we could scale it up. Like, so we know that in other domains of AI, like if you compare GPT-4 to GPT-3 and GPT-2, when you scale those models and provide them with more compute and more data, they kind of like really improve and can sometimes have entirely new capabilities that the smaller models don't have. So I think there is kind of like this open question to what extent this will also work for protein language models, and I think it will be interesting to see. Again, judging from other domains of AI, I'd be surprised if we don't see this kind of like similar scaling as we scale those models, but it's something that we're still waiting to see. And also, I think there is kind of like the big challenge of finding some analogous approach that will work for non-coding effects, right? So protein language models can only deal with coding effects, which is great. Those are like very important effects, and we care a lot about them. But we know that for most diseases and traits, actually most of the phenotypic variance is in non-coding effects. So for example, people have tried to do the same with DNA sequences and training DNA language models, which so far haven't worked very well. But I think it's mostly technical challenges, and it's difficult to predict what will happen, but I think there is a solid chance that we'll be able to get them to work.
Okay,